डॉक्टर साहब आपसे एक रिक्वेस्ट थी
जी।
जब आप अपना ये शुरू करोगे तो शुरू में
it's not there is some problem. It is not.
You can start, and let me just put everyone on a mute so it won't be a disturbance. Okay, just give me a second, and then after you can unmute yourself and you can start it.
Yeah, that's right.
And Professor Jay, you can unmute yourself and then you can start a lecture. Professor Bhatnagarji, please unmute yourself. Yes.
I unmuted already. Yes.
Yes, you can start a lecture. That's fine. Now everything is set up, so we can start. And you can also share. You already have a co-host, so you can share.
I'm not able to share my screen, the lecture itself, so that I can also show and talk about it. But if you have already gone through it, actually in the first lecture I have discussed about the DNA and gene, their structure. And if you are all aware, or if you have observed my lecture, the DNA is actually a molecule which is present in every cell, which is in our body. Well, in addition to that, it is present in bacteria, it is present in virus, it is present in all animals. Even a small animal, yeah, sure, please.
नमस्कार सर।
हाँ नमस्कार। आवाज नहीं आ रही है, आप वापस म्यूट हो गया आपका।
No, okay. So, however, I already muted everyone because this one is the lecture we have first started, right? So, after the lecture, we can open it for those the question answer session. So,
okay, it's not a प्रोफेसर जी, लास्ट वीक हम लोगों ने आपका पहला लेक्चर तो नहीं आया था, सुन नहीं पाए थे क्योंकि
अच्छा मुझे नहीं पता, नहीं लास्ट वीक जो इंट्रोडक्शन हुआ था उसमें मैं तो बाहर था, तो मैं एक्चुअली अटेंड नहीं कर पाया था। और मुझे तो अभी डॉक्टर कच्छारा ने बताया कि आपका लेक्चर लोगों ने पिछले एक वीक से देख रहे हैं, इसलिए आज शायद मुझे डिस्कस करना था। अगर आपने नहीं देखा है तो ठीक है, मैं नेक्स्ट टाइम डिस्कस कर सकता हूँ, ऐसा कुछ नहीं है। आप जैसा चाहें वैसा कर दूंगा।
Professor Ji, whatever we did it, we already posted out your lectures in lecture videos in our Google Classrooms. Okay, so Hansa Ji, you can go in a Google Classroom and watch those the videos.
Okay, okay, thank you.
Okay.
आप सभी हिंदी चाहेंगे या अंग्रेजी मिक्स चाहेंगे, कैसे चाहेंगे?
We prefer English, majority in English one, so at least it will be available for future generations as well.
All right, fine. Yeah, it's okay. So, as I said, the DNA is a macromolecule. Macro means large and present in every cell. Every cell means a small cell like a bacteria, which is a single cell. Then any animal you call like a sponge, which is found in sea and lakes, it's a multicellular. Then you can see some other animals, insects, amphibians, birds, human being. Every body is made up of cells. We are multicellular animals because we have a large number of cells. So every cell of a body consists of this structure, which is called as the DNA. We also say that DNA is basically a molecule which is responsible for all the activities of that particular cell. In bacteria, this DNA has little different structure, while in our case, it has a different structure. Okay. DNA is basically present in two ways. One, a linear structure. which is present in higher animals. Linear means like a strand. In the case of a single cell animals like bacteria, it is like circular structure. Okay. This is a basic difference in this. Now, in higher animals, the DNA is double-stranded. Double-stranded means there are two strands, strand A and strand B. However, in case of certain bacteria, this is single-stranded and in some cases double-stranded. Okay. Virus also has a DNA which is either single-stranded or double-stranded. Now, what these two strands do here? Actually, when the structure was first time showed, this double-stranded structure has an imaginary axis. There's no axis exist, but if you have an imaginary axis, this DNA is spirally coiled along this axis. Okay. The one coil, complete one coil, consists of 10 bars, and these bars are basically known as nucleotides. These bars are known as nucleotides. Okay. Now, what is nucleotide basically? So in DNA, these nucleotides which are present here or constitute the DNA, they are basically made up of three things. One, nitrogenous base. Nitrogenous base means it is a structure which consists of nitrogen molecule. Okay. The another substance which is present here is known as sugar. And the sugar is recognized as a ribose sugar. All right. And then there is a group called phosphate, PO4. In every nucleotide, it is made up of a nitrogenous base, a sugar, and a phosphate. Now there is a catch here. There are actually 4 types of nitrogenous bases, whether it is a bacterial DNA or whether it is a higher animal DNA. These 4 nitrogenous bases are adenine, thymine, guanine, and cytosine. And they are basically referred as A, G, T, and C. All right. Now, what happens in the nucleotide? If we just remove the phosphate group and talk about the nitrogenous base and the sugar, then it will be called as nucleoside. But when the phosphate is added, it is a complete nucleotide. Okay. Now the entire strand, which is made up here, consists of 2 nitrogenous bases actually. The one strand, as I said, when it is inspired, it forms a coil. So if you see the entire coil, as I said, there are 10 bars and each bar is referred as a nucleotide. So all these bars are made up of either of a pair of nitrogenous bases, and they are paired in a special way. Like the A or adenine always pair with thymine or T, A, T, and guanine or G always paired with C, that is the cytosine. All right. Now, A, G, and TC, whatever structure of DNA, always you will find these bases, and only these bases, 4 bases, constitute the entire DNA. They will be repeated several times. All right. When you see the diagram in my lecture, then you can imagine that how they are arranged in the picture. So A, G, T, C, but it is not necessary. Sometimes it could be T, A, or C, G. So in a one strand, if you see one strand of a DNA, A, sugar, G, sugar, there may be again A, sugar, there may be T, sugar. Why I say this? Because the 2 nucleosides, as I said, they join together with a group called phosphate bond. Okay, so that's why that phosphate is present. All right, so there will be 10 nucleosides, but there will be 5 phosphate groups on both sides. And these phosphate groups are basically joining this ladder structure. Okay. This is basically a structure of a DNA, a simple way. Now I will talk about the gene structure because it is based on a DNA. Say DNA could be a very long structure. If you take out a DNA from a cell, it is always present inside a nucleus. It is a circular structure which is bounded by a membrane. But this type of structure found in case of higher animals only. So if you see any cell under a microscope, you will find a large circular structure or elliptical structure or some wavy structure. And in the center, there is a circular structure, which is basically called as a nucleus. The DNA, entire DNA present inside this. Okay. This DNA, which is present in a nucleus for a particular species, is known as a genome. The word is used as genome. Okay. Now, in the case of a bacteria, there is no boundary or there is no membrane outside. So the DNA of a bacteria is always free. And interestingly, there are two types of bacteria, two types of DNA. There is a linear DNA and a circular DNA. And these circular DNA are characteristic of each bacteria. All right. Now, I already explained the structure of a DNA. And now I want to tell you that DNA sometimes in some animals, the DNA such could be so large that if you spread it linearly, it may go from here to the moon. The length is so big, you cannot just estimate it, how big it is in such a small nucleus. Okay. Such a big length. And this entire DNA, the full length, is made up of only 4 types of nucleotides, A, G, T, and C. They will always repeat it right from the beginning to the end of a DNA strand. Okay. Now, another thing here. This linear DNA, once we have taken out and if we spread it, then on this DNA, genes are present. Now, question is, what is gene basically? So gene is nothing, but it is a part of a DNA, which is also made up of the same structure. A particular area on a DNA, which is active and has some functional property. Functional property, I will explain later. What kind of functional property is basically called as a gene. So if suppose we have a DNA, which is about 20,000 nucleotides. All right. If we have a DNA which has 20,000 nucleotides, just for imagine, in that case, a gene could be made up of 5 nucleotides to 200 nucleotides. 5 nucleotides to 200 nucleotides. Okay, it's not more than that. Basically, the largest gene is about 200 nucleotides. And this structure, which is called as gene, or a structure made up of 200 nucleotides, has functional properties. Now, what kind of functional properties it has? See, I will explain different way. We have a muscles, and muscles are made up of proteins, right? Large number of proteins are there because sometimes there is a stretch, sometimes you have a cramp, sometimes your muscle moves. So what is this? This is because of the proteins which are present there. If I explain you that the entire structure of a cell is made up of about more than lakhs of type of proteins, then you will surprise, right? Lakhs of type of proteins. And these proteins are basically synthesized in the cell itself, in such a small cell, which you can't see with your eyes. And you have to use a lens or a microscope to see it. Okay. Now, what protein is doing here and how it is synthesized in a cell? Well, the protein is basically synthesized by this gene. The gene is responsible. Gene is controller. Gene controls how protein will be synthesized. Okay. Now, again, we have to understand here some few things. protein function in different way. Like you have studied that you know that in the stomach there is a digestion of a food, different type of food is digested. You also know that in saliva in our mouth, which is secreted, food is also digested there. So how this food is digested? It is digested by the action of enzymes. Like there is an enzyme called lipase. There is an enzyme called pepsin, which is present in our stomach. Okay, simple lipase is present in our saliva. Pepsin actually digests the proteins. Lipase digests the fatty acids or lipids. Okay, something like this. So different type of enzymes are present. This is the one way the protein act in our body. The other way, you have heard that we have different kind of glands like thyroid gland. बहुत सौ का प्रॉब्लम हुई होगी थायराइड की. तो थायराइड की प्रॉब्लम कैसे होती है? जब हार्मोन कम हो जाता है, ज्यादा हो जाता है ना? थायरोटॉक्सिन हार्मोन which is secreted from thyroid gland. There are other gland known as a pituitary. Growth hormone secrete होता है. तो early birth के time पे when the growth hormone is secreted, it is helping in the growth of a body. बच्चे में ये ज्यादा secrete होता है. So this type of a protein is known as a hormone. Growth hormone, thyroid hormone, oxytocin, which is present in the estrogen, which is secreted in the females by the ovary, like testosterone secreted in the case of a male by testes. They are all proteins. And interestingly, all these type of proteins are synthesized in that particular cell, which is so small. Okay. I have given the example of enzymes, I have given the examples of hormones. But what about our structure? Cell structure. Do you know that every day, large number of cells of your skin, they are removed. Every day they are removed and new cells are formed. Do you know that large number of RBCs get digested? They become dead and new RBC are formed. Do you know that large number of cells on the skin, large number of cells in the brain, they get die after some time and new cells are produced. So in body, this type of functioning is going on. New cell, earlier cells die, new cells are produced. So how these new cells are produced? This structure of a cell is made up of protein. Again, I'm not going into details because that will be another lecture completely. But what I can say is the cell structure is made up of large amount of protein, fats, and carbohydrate. But major contribution is protein. So this protein, which is responsible for the structure formation, is known as a structure protein. The hormone responsible for controlling our body activities. And then third, the enzymes responsible for digestion and various other activities. For example, you get the energy. Do you know that there is a full cycle known as glycolysis, which is consists of large number of enzymes, and they are using glucose molecule which we eat food. The glucose is taken and then it is used to produce the energy. So how that energy is produced from a glucose molecule? By the action of a large number of enzymes. There are steps, a cycle known as a glycolysis. So there are basically three types of proteins. Now the question is how the cell decides that now we have to produce the hormone which is required in the body. Now how the cell decide that a protein is required for the formation of a cell, and how the cell decide that an enzyme is required for the digestion, right? So basically what happens, the gene is basically responsible for this. And do you know that the entire information for synthesizing a structure protein, a hormone, and what you call this enzyme lies inside this structure, which is basically called as a gene. And again, if I say the entire information lies in these four magical words A, G, T, and C. Okay. Now, here we have to also understand one thing. Now it is a complicated part, so you have to be careful. A protein is made up of amino acids. There are 20 different types of amino acids in our body. All these amino acids are utilized in our body for synthesizing different type of proteins. Okay? आपने हरगोविंद खुराना का नाम सुना है. Professor Hargovind Khurana was given Nobel Prize first time to elucidate this phenomenon, that how protein or a gene is responsible for synthesizing the protein. Okay. Now question is how gene is controlling. And as I said, it is a complicated. I said already that on a DNA, A, G, T, C is not necessarily in the same sequence. There may be A, there may be T, there may be again A, then C, then G, or G, 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 then AA, something like this. So on entire strand, they are distributed like this. Okay, on both the strands. If there is a A, तो उसके सामने वाले पे T होगा. अगर उधर A है, तो इधर T होगा. Something like this. Okay. Now, what happens when the DNA is ready to, or gene recognize, or there is a signal to the gene that a hormone has to be synthesized? In that case, the gene get activated. और इस जीन के ऊपर एक सिंगल स्ट्रैंडेड स्ट्रक्चर बनता है, जिसको हम कहते हैं RNA, राइबोन्यूक्लिक एसिड. DNA is basically called as deoxyribonucleic acid. अब राइबोन्यूक्लिक एसिड वैसे तो it is exactly similar to DNA. Only difference is RNA is basically a single stranded structure, but DNA is a double stranded structure. Okay. Now, since it is a single stranded structure, still it consists of A, T, G, C. But there is a catch here. Here, T is replaced with another nitrogenous base known as uracil. And the word used for it is U. Okay. The word used it for T is, to replace T is U. So if in any structure, single linear structure, if you find A, U, G, and C, then it is not a DNA, it is a RNA, ribonucleic acid. And this ribonucleic acid, which is formed on DNA, basically carry information for a particular protein. Now you should also know here one thing. How we say that this protein is a hormone, this protein is a lysoenzyme, and this protein is a structure protein. How we recognize it? Well, we recognize it in this way. As I said, every protein is made up of amino acid, and 20 amino acids are there. They are also arranged in a sequence in a protein. There are three types of proteins: simple protein, secondary protein, and tertiary protein. Single protein consists of single chain of amino acids. That could be a 10 amino acid, 20 amino acid, 100 amino acid, 500 amino acids. Okay. And same thing in all these structures. Structure protein, hormone, or enzyme. Same thing. Okay. Now, one thing is there. A protein should have a specific number of amino acid. It may be 20 amino acid which are present there, or they are repeated again and again. If suppose a protein consists of a 50 amino acid. So these 20 amino acid will be repeated twice, thrice. They will bind in the chain. All right. And this way they form a complete length of the protein structure. Now, if there is a secondary protein, there are more than 2 chains, right? And these chains are combined together or bind together with sulfur bonds. There are some structures which bind them, and that are known as sulfur bonds. Okay. Tertiary proteins are actually consist of chains, but the chain is folded. and make a complex structure. And that's why they are called as a tertiary structure. But they are all synthesized in a similar way. Here I also want to tell you that if there is a single chain in a protein, that will be synthesized at a time. But if there are 3 chains in a protein, they will be synthesized simultaneously. If there are 20 amino acid in a chain, protein chain, all 20 chains will be synthesized simultaneously. Okay, and the synthesis basically takes place on this RNA, which consists of or carry the information from DNA. Okay, so basically the information for a protein to be synthesized. Now let us consider we are synthesizing a protein in a cell which consists of 20 amino acids. Okay, or 5 amino acids. It is better to understand, right? 5 amino acids. So, and what is this 5 amino acid protein? Suppose hormone, and it consists of specific 5 amino acids. I can name 5 amino acids. Like one is, you can write down so that you can understand. One is alanine, another is valine, third is glutamine, fourth is isoleucine, and then methionine. Alanine, valine, glutamine, isoleucine, and methionine. So these are the 5 amino acids. Now they are in a particular chain and they are arranged in a sequence. That first is alanine, second is valine, third is glutamine, fourth is isoleucine, and fifth is methionine. If suppose they are synthesized by RNA in this way, let us recognize it as a hormone, or suppose thyroid hormone. Okay, but if you change the sequence, like if methionine comes first, then there is a glutamine, then alanine, then valine, and then isoleucine. So the sequence is little change. Okay. Now this will be a different substance. It may be a thyroid hormone. No, it will not be a thyroid hormone. It will be a growth hormone, or it may be some enzyme. Okay. So the nature of a protein depend on the sequence of amino acids. There is another point also. If suppose I add 2 more amino acid in a chain, that methionine, glutamine, alanine, valine, isoleucine, and now I add threonine, and I add 1 more like cysteine. So now what happens? Here becomes this 7 amino acids. This will have an entirely different nature. It may be an enzyme. So a nature of a protein always depend on 3 things. 1, the number of amino acid in a chain, type of amino acid in a chain, and third is their arrangement in a chain. I repeat it. The nature of a particular protein depend on the number of amino acid present in a chain, number of a type of amino acid present in a chain, and their arrangement in a chain or their place in a chain. If you change these, suppose the number is 5, if you made it 7, the things are different. If suppose the arrangement is changed, the things are different. Okay, so basically these 3 things are responsible for the characters of a particular protein, that what it is. All these 3 informations are transferred from DNA to RNA in the form of nitrogenous bases. Okay, in the form of nitrogenous bases, right? Now, again, a little complicated thing, but I will try to explain it very easily. Let us make a structure of a RNA. You can also write down simultaneously, draw a single line, and mark it at several places about 10 marks are there at a little difference. At the first mark, you write down A. At the second mark, you write down U. At the third, you write C. At fourth, you again write U. Fifth, again you write A. And sixth, again you write A. And again you write U. And write C. So it is this a kind of a sequence in an RNA. Consists of 1, 2, 3, 4, 5, 6, 7, and 8 nucleotides. Okay. I repeat again. First is A, second is U, third is C, again U, A, U, and C. Okay. Shall I go forward? Now, if you make a pair means 3 nucleotides, like AUC. Okay, AUC. Similarly, you can have UAA. And let us add one more, so that will be easier. So last you can write down C again. So last will be UCC. Okay, so again there will be a 3 in a pair, 3 nucleotides. So we have here basically 3 groups. First consists of AUC, another consists of UAA, and third consists of UCC. Okay. Now what happens basically in our system, these three nucleotides are basically called as code. And this was first time suggested by Hargobind Khurana. These are called as code. And actually they are referred as genetic code because they are related with the gene or they are present on a gene. So we call them genetic code. Okay. And each of these 3 nucleotides in a pair recognize one amino acid. Recognize one specific amino acid. Okay. However, there are more than 64 type of codons. 64 type of, because what happens if suppose you make these pairs, this is a random, okay? It's not necessary that it is present exactly like on your DNA or your gene. It is a random. I'm just forming it to explain you. Okay. So what happens? Suppose one AUC identify a amino acid called as methionine. Okay. AUC identify methionine. UAA identify glutamine. Now what happens here? The last code, genetic code, like UCC, there is on the extreme one end of a RNA, there is always one genetic code which not recognize any of the amino acid out of these 20. This is purposefully there because if they don't recognize anything, the growing chain will stop there. Okay, so out of 64, there are 4 that kind of genetic code which do not recognize any amino acid. Okay. And that's why what happens when the chain is growing and amino acids are incorporated in the chain. And finally, once this type of a what you call the genetic code comes, the chain immediately stops there. And that is a natural system on every what you call RNA form. These are basically called as terminator codons. They are known as terminator codons. Okay. Now let us see, understand here how the chain is basically formed. Now the RNA is formed from the DNA, and we have already formed that RNA here, which is consisting of 3 different kind of what you call nucleotide pairs. When the chain or synthesis has to begin, that time this RNA comes in contact with another structure which is present freely in our cell nucleus or outside the nucleus. And these structures are spherical structures called as ribosomes. Okay, these are called as ribosomes, and these are basically the structures where the synthesis takes place. Where the synthesis takes place. So what happens? The RNA complete strand can be written like this. On the end where AUC is present, you can write down 3 prime, and where UCC you have written, you can write down 5 prime. 3 prime and 5 prime, or the beginning end and the terminating end. Okay. Why 3 and 5? It will be again very difficult to explain you, because if you have a biology background, then I can explain very easily. Otherwise, it will take a long time. But we can manage it to explain you by a small another figure, and that I will explain in that way. Okay, I will send it so they will distribute you. Now, what happens? The synthesis began from 3 prime end, where AUC is there. The ribosome will enter on this RNA structure exactly in a similar way. If suppose we have a cotton strand, एक धागा हो हमारे पास, और उसमें एक बॉल को हम पिरो दें, उस बॉल को एक तरफ से धक्का दें, the ball will run and pass out from the another end, right? The ribosome moves on RNA in a similar fashion. It enters from the one end and pass on from the another end. So during the movement, it will recognize these 3 pairs. Okay. For example, when it enters, comes to the AUC, it recognizes all these 3 structures. Now the ribosome is present of AUC, so it is activating AUC. What happens? As I said, that the AUC is controlling methionine amino acid, or responsible for methionine amino acid. So the methionine will bind or start the chain, right? The methionine will come and occupy site on the ribosome. So the first amino acid is incorporated. Now, meanwhile, the ribosome is moving forward, and now it will come to the next. So the next is UAA, and I said that the UAA recognizes glutamine. Okay. So when ribosome comes in front of a glutamine, a UAA, the glutamine will come and bind to methionine. There is a binding between these 2 amino acids known as peptide bond. Known as peptide bond. Now, meanwhile, the ribosome is moving again, and it will be in the presence of another 3 pair of codons. Now, another amino acid will join, then ribosome will move again, and another amino acid will join. So like this, amino acids will go on joining. Another catch is, as I said earlier, that when the synthesis takes place in a protein, there is a single chain of amino acid, then it will synthesize by 1 ribosome. But if there are more than 2 chains, that time, that many ribosomes will enter. So the first ribosome entered, a chain starts, the second ribosome will enter, and then second chain will start, and then third ribosome will enter, and then the third chain will start. Like this, all 3, the first will move ahead, second will follow, and the third will follow. And like this, all the 3 chains simultaneously synthesized on the RNA. So in this way, a specific protein is synthesized. Once this is synthesized, the protein finally, when the ribosome comes to the terminator codon, as I said, which is UCC, in that case, since no amino acid is added into the chain, the synthesis stops here. So a protein is formed with a single chain or a double chain, and now this will be released from here, and it will go to the cell cytoplasm. Okay. Now there is a modification takes place inside, and then it will be recognized as either the hormone, or as a lysosome or enzyme, or as something, a structure protein or something else. And then it will be utilized properly. Okay. So in this way, basically, the protein is synthesized in a gene, and gene controls the synthesis of a particular protein in this way. So basic function of a gene is basically to synthesize a DNA, to synthesize a protein. Now you will say that you have heard that the gene is responsible for heredity, right? That's what you have heard. And that's why everybody say, "Sab, aapka jo naak hai, is tarah ka hai, uske liye gene responsible hai. Your eyes are black, that's why gene is responsible," right? So I will take an example of your eyes. I think you are a mother, Hansaji, and you know that when the child is born, that time the eyes, color of an eye is little brownish or little light grayish. But as the boy grow further, means after about a month, you will see that the color changes and it will become little dark. And finally, when the boy comes to suppose of 6 months age, the eye color will become completely black. You might have also seen the color of eye like blue. You might have also seen color of eye like brown. So how these different type of structures are there, or different type of colors are there in the eye? Somebody has a gray color, somebody has a blue color, somebody has a what you call brown color, and somebody has some other color, mixed color. Even the green color eyes are also found in some cases. It is rare, but it is also reported. So question is how this variation is there. This variation is a heredity, right? How gene controls? The gene is responsible for formation of the brown color eye, the green color eye, the blue color eye, and the gray color eye. And what they are basically proteins. Okay. So basically the gene is responsible for these type of activities in our body. I have a gray color because the protein, the black protein, which is required for blackening of a hair, is not produced in me. That is again a genetic, because my father had the same case at the age of 35. He had a complete white hairs, and at the age of about 38, I also had the complete white hairs. Which is controlling it? I received it from my father. And who is responsible? The gene is responsible. So in this way, gene controls everything. Okay, I think that will be enough. You can ask some questions if you are interested.
So if we want to change something, then you have to change that particular sequence of gene or replace some
Yes, that's right. That's
amino
right.
acid or something.
Say, I will give you an example. Long back in about 1989-90, there was a boy. He was born in England. His name was Bob Hope.
Yes.
And now he dies. But what happened? Bob has one particular disease called as SID, SCID, Severe Combined Immunodeficiency Syndrome. That was recognized first time in England. This disease. What was this disease? Disease was this boy was actually placed in a plastic bubble inside, completely not connected with outside environment. The reason being, he was able to get every kind of infection which is possible. So doctors were surprised that what is happening. And in that time, ultimately it was known when they tested him and tried to find out genetically. What they found is a particular gene which is responsible for an enzyme is absent in him. Right from the birth, it was absent. And that gene was known as ADA gene, adenine diamynase gene. It is a hormone, enzyme, which is responsible for production of a type of protein which develops or responsible for the immunity so that you don't get infection. So that gene was completely absent. Now that time there was no treatment for this particular disease. The boy was kept in a bubble for a long time, about 15 years. And there was no treatment the doctors or the scientists were able to develop for this, because how to approach the gene, how to introduce the gene. Now the things are possible today. If the gene is absent, a gene from outside can be inserted into a DNA, a functional gene, okay, which is now possible, but that time it was not possible. It is also possible that if you have certain particular gene which is causing a disease, it is possible that you can cut the DNA and take out that gene and replace it with a functional gene. These techniques are now available, but still not in a regular use, because scientists are not yet confirmed that if we introduce a particular gene, how it will perform in the body. So still experiments are going on, but the future is very near. Maybe very soon we will have this kind of a treatment where the gene are completely replaced. A disease gene or a defective gene can be replaced easily. Okay. Any other question?
Yes, Professor, I have a question.
Yeah, sure.
If there is somebody has a behavioral or a mental disease, can you alter the gene to correct the mental disease?
Yes, it's possible.
Is it done at the moment anywhere?
Not yet done, but it was attempted in a different way. Not anybody has tried, but actually attempts are made in the case of animals. Okay. But in human being, this kind of experiment is not yet, because unless it is approved by the government, these kind of experiments cannot be performed, or these kind of surgeries cannot be performed in the human beings. Okay, but
because
it is possible. But these days there is another way of doing it. If suppose somebody has aggressive behavior, it was found that yes, gene is responsible for it, but there are some different kind of treatments also possible by like increasing some kind of a micronutrients in the diet. Like in Germany, somebody was working. I also worked with him, Professor Novikov. He was working on this kind of a project that if suppose we increase some micronutrients like nickel, magnesium, or something like this in the diet, you can change the behavior. Like you can make the aggressive person, and if you reduce it, you can reduce the aggressive behavior. That experiment was for the military. Okay. So yeah, earlier that was done, and these kind of study, I said that the experiments are going on, but still gene therapy is not in what you call normal use. So once it is approved completely, then the gene therapy will start. But you know that these days large number of medicines based on DNA and RNA are also given. In the case of a cancer, breast cancer, especially, there is a DNA vaccine or DNA medicine which is given to the patients, or RNA medicines given to the patients. Yeah. So the work is going on.
Okay. Because here in the UK, the mental illnesses have just escalated so much.
Yeah, I know that.
So I was just wondering whether anything's been done about
possible. It is possible, but still the therapy is not yet what you call open for all.
Okay, thank you.
And that too is, it's very expensive. Very expensive. It's not that easy.
Okay, thanks.
Thank you, Professor. Yeah, I have one question.
Yes.
Yeah, thank you for a wonderful talk. I just wanted to know whether can we connect this karmas and this karma and gene?
Yeah, that's right. Now, I will give you one example.
Yes.
My father was basically a teacher. Well, he retired as a senior director of education, but initially he started as a career as a teacher in a school.
Right.
And I tell you, he was a man of geography. Okay. And he has a wonderful artistic attitude. He can draw anything. He was able to draw. You give him a picture and he was able to draw it.
Okay.
Now the second thing which I'm connecting with this, I can draw anything. You give me any picture, I will draw it. Exact same. But I never learned it. How I got it must be transferred from my father.
Okay.
Right? So yes, these karmas, whatever you say, they are all connected with the genes. The influence of
Can
gene
I
is
get
there.
for more information? Yes. Can I
Yeah.
get more information? Can you suggest any book on this?
Yeah, sure. You can write down by email m.mahib.
1 minute, sir. 1 minute.
M.
Dot.
Mahib, M-A-H-E-E-P. M-A-H-E-E-P.
Yeah,
Mahib
yeah, Mahib. Yeah.
@gmail.com.
Right, sir.
So you can send your questions on that and I will answer it. Okay.
Right. Thank you. Very nice of
And
you, sir. Thank
any
you very much.
information which I have, I will transfer it.
Dr. Saab,
Okay.
Yes, sir.
Thank you.
this is Alok Jain. I have a question for you.
Sorry.
I have a question for you.
Can you be a little loud? I'm not able to understand what you're saying.
Can you hear me now?
Yeah.
So could you compare for me, let's say, a genetic treatment versus a conventional medical drug treatment? In dono, how does a drug treatment work? You explained how the genetic treatment works.
Yeah. It's very clear. See, in case of a normal treatment, a doctor gives you a medicine,
Mhm.
right? This medicine is actually a simple chemical. Now the chemical is basically, again, I tell you, it's a synthesized chemical, synthesized in a laboratory or by a industry. But this chemical is based on certain plant product. Like, you know that violet grapes,
Yeah,
I'm not talking about the green grapes, I'm talking about the black grapes.
yeah.
This black grape consists of a substance called resveratrol. Another example is a quinine, which is also from a plant. Now, these days, quinine is synthesized in the laboratory. It's not obtaining from the plant because we know the structure of the quinine. Okay, so this is basically a chemical. But when you talk about the genetic therapy or gene therapy, it is basically a gene which is given to, suppose, a person. The gene will be introduced. A group of nucleotide sequence will be introduced into your DNA. The chemical will act on DNA and produces influence and ask gene to act again like that. But the gene is basically introduced in the sequence, DNA sequence or DNA strand, and then it will function according to the requirement. So there is a difference in the chemical reactions and there is a difference in gene therapy. Okay.
Thank you, doctor. Thank you.
Jai Janendra Professor Ji, my name is Shobha.
Yeah.
I want to know, does meditation in any way, like it has influence on immune factors? It modifies the good ones and the bad ones. Do you think that it can cause any modification in the genes also? Has there been any work done so far? Meditation and gene?
There are people working on it. There are people working on it, try to understand the connection, possible connection between these. But as such, the final has not come yet. People believe, some scientists believe in it, that this is possible, but the final conclusion has not come yet. But I personally also believe that, yes, there is a possibility of a change in a gene functioning.
Because
Right.
after all, if immune factors can be changed, rise or decrease,
I believe
after all, the basis is only protein, and gene is also the basis is protein. And with
that's right. Right. Yeah.
Thank you very much.
Okay.
Dr. Saab, I had another
So
question.
are we done for today?
One more question for you. One more question.
Yeah,
for
sure.
you.
Yeah.
So when they say cell mutation has occurred, what does that mean?
Mutation. Okay. That's right. Mutation means what happens if suppose you are walking on a street, right? And as we say that the sunlight which falls on you consists of ultraviolet rays.
Hmm.
Okay. Sometimes the doctors, which are physiotherapists, they expose you with the ultraviolet light to treat your pain. If suppose there is a long-term exposure to X-ray, to ultraviolet light, in that case, these two rays are actually suggested that they affect the gene.
Hmm.
They can alter the structure of a gene. Alter means the word used mutation. So what happens? Either there is a breakdown in a sequence on one of the strand. So this is known as a nick, N-I-C-K, means a small nick is there and that the strand is breakdown. That is one way. And actual mutation is actually in different way. What happens here? Either a sequence is reversed, suppose 3 nucleotides, A, T, C, and they become CTA, or this particular CTA is deleted, or a new addition is there. So these kind of changes in a DNA, because of long-term exposure of X-ray, long-term exposure of ultraviolet light, are basically called as mutations.
Yeah, thank you. Thank you, Dr. Thank you.
So the question for other people is the people who gets the Down syndrome, who gets the 45 chromosome, it's the same thing happens?
It's not basically a mutation for Down syndrome. It's something else. It's not because of a mutation. It is something else.
But
That has to be explained in a complete lecture.
okay, because
But very basically, I tell you what happens when the ova is formed.
Yes.
Okay. And then there is a fertilization between sperm and ova. And after that fertilization, a cell is formed known as a zygote. And then it undergoes division to form a child.
Right.
That time what happens? Certain chromosomes don't separate.
Okay.
They goes along with. Basically what happens, every cell should have 46 chromosomes.
Right.
23, 23. 23 comes from father, 23 comes from mother.
Right.
And actually, when the child is formed, zygote is formed, and then there is a division. Again, there is a division of a chromosomes. So what happens when there's a division of a chromosome? Some X chromosomes goes to a one cell, and some the remaining goes to a one cell. So these extra chromosomes are actually responsible for formation of these kind of syndromes. Okay, or sometimes some less chromosomes are responsible for formation of these kind of syndromes.
Right. Thank you.
Either more or less.
Thank you.
It is not a mutation, but this kind of a phenomenon is not a mutation.
So
okay.
Sometimes it happens with the radiation. If somebody is exposed to the radiation, they can also develop Down syndrome.
Radiation Down syndrome, I have not heard it. I don't know.
One of the reasons in hospital they give you X-ray badge for
No, I don't think so.
This only happens.
But I will answer you. Okay, you contact me on my email. I will answer you this.
Thank you.
Dr. Saab, can I ask one more question?
Yeah, sure.
The question is, we talked about genetic treatment. We talked about drug treatment. And then there is this issue of when people meditate, for example, through meditation, there is secretion of certain hormones. At least that's what the theory behind the meditative treatment is. How do you explain that in terms of genetics?
Can you repeat just briefly?
So how do you explain the positive effects of meditation on human health in terms of genetics?
See, as I already said, that people are studying it and try to see that different kind of meditations, how it affecting the person's behavior and how that change in behavior is actually affecting the gene itself. But as I said already, there is no conclusive evidence is there still. That is the problem. For example, one student has done a thesis under me also, and we studied about 4 different kind of yoga postures and their effects. But final conclusion is very difficult, I tell you, very difficult. It's not that easy to understand it.
I see.
But I will give you some examples again on the email,
Okay.
Okay. so that
Thank
you
you.
can read and understand it.
Thank you, sir. Thank you.
Thank you. Very nice.
Okay.
Thank you.
So are we done for today? And the remaining question you can ask on email and I will answer it. Okay. That will be better because I can able to attach some extra information also
Yeah.
on
Thank
the
you.
email.
Thank you.
Okay.
Thank you.
very much.
Thank you very much, sir. Jai Jane.
Thank you, sir.
Thank
Jai Jinendra.
It was okay. The lecture was clear.
Very
Yes.
good.
Yes.
Thank you.
Yes, thank you.
Very nice.
Thank you very
Thank
much.
you.
Thank you, Professor. Jai Jinendra.
Thank you.
Jai Jinendra. Hmm? It's okay. People are still. They will do it themselves.